Good morning, everyone. Welcome to TeleShadowing. We're live on YouTube. Please feel free to ask questions in the chat box as we go along, and they'll be addressed. I'm honored to introduce our mentor for today, Dr. Spellicy. Dr. Spellicy completed her Ph.D. in neuroscience from the University of Georgia. During this time, she worked as a graduate research assistant studying the therapeutic potential of neural stem cell-derived extracellular vesicles in stroke and other neurodegenerative conditions. This work resulted in an Excellence in Research Award awarded to one student in the life sciences by the graduate school, as well as 8 publications, 5 oral conference presentations, 5 invited talks, and 29 poster presentations. Dr. Spellicy is currently completing her MD and matched for Neurological Surgery Residency. Now I'd like to request Dr. Spellicy to begin today's session. Thank you.
Great, thank you so much for that great introduction. And you crushed saying neural stem cell derived extracellular vesicles. I know that's a mouthful. So yeah, let me get my slides up here. Thanks for the invitation, and thanks to everyone who's kind of tuning in this morning. I know it's a Saturday morning, so really appreciate everyone being here. But yeah, as she said, my name is Sam Spellicy. I'm an MD PhD student. And so in the talk today, I'm kind of going to talk about the first half as kind of my path and a little about MD PhD programs and a little bit about kind of everything I wish I would have known in undergrad, honestly. And then the second half is going to be more about kind of the research and the neurosurgery residency and a little bit about neurosurgery. So yeah, if you don't want to hear the first half and think kind of the pathway stuff is boring, just come back in 30 minutes and then it'll be all the science stuff. So, okay. So this is the first half, things I wish I knew in undergrad. So I grew up kind of in Prince George's County, so right down the street from Maryland. I don't know if anyone has seen this documentary, but it is about Prince George's County specifically. And so not a lot of counties have a whole documentary that Kevin Durant made about them. So I always like to include this. But yeah, so I grew up kind of really local to Maryland and just didn't have a lot of science and medicine kind of experience or exposure before I came to school. So I wasn't kind of set on this kind of MD, PhD or PhD path actually when I started at Maryland. So this is kind of a little example of my timeline when I started at Maryland. So I started actually undecided major, didn't know what I wanted to do. After a year or so, I declared journalism in government and politics. And I just really liked the DC area. I thought it was kind of cool to be doing government politics so close to DC, and I liked writing, so I thought it made sense. So I kind of was going down that path. And then I got a B in grammar for journalism, and I was like, nope, not for me. Orgo's got to be easier than this. So then I changed to physiology and neurobiology because I was like, it just wasn't for me. I'm not great at grammar, apparently. So I switched over and it was really kind of the whole time I had really been interested in science. And I guess it took kind of a push from someone to say, like, if you really like science and medicine, why don't you just do it? I think no one had kind of even pushed me to say that that was an option for myself. So I kind of started my, switched over my major. And then I started kind of my junior year, started doing research. So you know, if you're interested in PhD or pre-med, you know, obviously it wasn't a completely straight direct path for me. So I didn't even start research till kind of the end of my undergrad. So if you're interested, there's still time to start and get involved. I worked with Dr. Karen Carlton, who I think is still at Maryland. She teaches a couple classes there. So if anyone's kind of had a class with her, she's great. She's a great mentor. Really, I think what this experience did for me, besides exposing me to research, but it just kind of exposed me to the hierarchy of a lab and how grad school kind of works. So I still remember going into the lab on the first day and someone introducing themselves as a postdoc and then someone introducing themselves as a grad student. And I just really didn't know what those terms meant. I didn't know, I hadn't been exposed to it. I didn't know, like, okay, who's what's a postdoc? And so for anyone who doesn't know, usually grad school, after undergrad, people start grad school. You can be any gap of time between when you start your grad school, but you can do a master's or a PhD. So a master's is usually kind of a 2-year program. And then a PhD is kind of like a 4, 5, 6, 7-year program. And so usually you're doing the same type of work, you just have a smaller project if you're doing a master's, something that could be completed in 2 years. If you're doing a PhD, it's kind of a larger project that might take 4 years to complete, but you're kind of doing similar coursework at the beginning. And then a postdoc is someone who's finished the PhD and is now still working in the lab as a PhD scientist, but they don't have their own lab yet. So it's kind of like a stepping stone to then one day having your own lab, like Dr. Karen Carlton. So she probably did a postdoc and then started her own lab at Maryland. So after learning all that and kind of learning how labs work and what the hierarchy was, then I decided I wanted to do a PhD myself. I really liked doing research. I liked being in the lab. I liked the environment. I didn't think that, undergrad was going to be enough for me. Like I enjoyed school a lot. So I felt like I wanted to keep going. I wasn't ready to kind of pick a career and settle down just yet. So then my senior year, I took a class that was more medically oriented. And this is at Maryland, that we would look at like neuroimaging of a patient. So say a brain, and there'd be a lesion on the brain, and you'd have to describe what the symptoms would be from that lesion. Or the reverse, where a patient would come in and they'd say, oh, this patient, just in a question stem, but this patient has a motor function on this side, deficit in their right lower leg, but then they have sensory issues in their left lower leg. Now you have to find out what's going wrong and where the lesion is. So pretty cool, very medically oriented class. And I think that's when I really realized, oh, wait, I like med school too. I like the MD. I like working with patients and working through problems that way. So that's when I decided I want to do an MD PhD. So you can see I was at my senior year. So it was kind of, a long journey to kind of come to even decide I wanted to do an MD PhD. So after, then I started a post-bac at NIH. So kind of in that gap between finishing undergrad at Maryland and before starting my MD PhD, I kind of did this post-bac. And so that's an option for people too, if you're still kind of figuring it out. And even if you're not, I think a post-bac or kind of a gap year is always a great option. So just as a little plug for the program at NIH, it's called the NIH Post-bac IRTA. So if you Google that, it'll pull it up. This is a QR code that'll actually take you to the application there. I just highly recommend this post-bac for anyone who's thinking about it or just wants to learn more. There's a YouTube about the program and what it entails, but pretty much you just get to work on NIH's campus with some of the best researchers in the whole world. So in Bethesda or in Rockville, and I was kind of based off the main campus. But you get to work with a bunch of students who are in the same boat as you. So students who are doing the same program. They finished undergrad, they're about to start grad school or med school, and they're just kind of fitting in something in that gap there. And so it's people your own age or who are in the same boat as you. You get to learn from each other, and then you get to learn from mentors who are kind of these world-renowned, famous scientists. It's a great experience. And then this is kind of just a picture of what the campus looks like. So this building in this picture is called Building 10, and that's kind of like the hospital center at NIH. So they do have clinical patients too. So if you're like me, where you are kind of interested in the clinical side and the research side, you can get research on the campus and clinical stuff. So it's a great environment. Highly recommend. And then just one little plug for gap years. Don't get bogged down by the actual data here. I pretty much just put this in to say people are looking at gap years and who are taking gap years, and there's a whole publication about this. And this publication just came out a couple months ago. But pretty much saying that a lot of people going into MD PhD programs are taking gap years. And what are they doing with that gap year? What do they do in a post-bac program? Who's taking the gap year? So this is just to show you that a lot of people are doing it. So if you had any reticence of saying, I don't know if that would make me look bad to do a gap year. I think back when I was applying, a lot of people thought a gap year, you only take it if you need to boost your application in some way, which I don't think that's the case at all. I think it's a great time to get more experience, get more lab experience before you go down this really long path, and then really take a time to take a break from school and do this self-reflective period to figure out, okay, is this actually what I want to do? Because it's a long journey. So you kind of want to answer that question before you get in it. And then a last little nod to this organization, APSA, the American Physician Scientists Association. So if you are interested in doing an MD, grad school, MD PhD, DO PhD, vet PhD, which is an option, this organization has people who are in those training process all come together, and they do webinars for undergrads to tell you about all of those programs. And so this kind of tweet in the middle here, it says session 10 applicant interactive series. So they are doing a series for undergrad applicants who are applying to MD or DO PhD programs, and there's 10 of these. So they have a different topic every time. You tune in, and this topic for this tweet specifically was about choosing a program and navigating waitlist. So if you are on a waitlist for a program, how do you choose from there? And then the earlier sessions were more about the early parts of the application process. So yeah, just wanted to put that on here just in case anyone's interested in learning more after the talk today. This is a great organization to find information about physician scientist programs. And here's a QR code that just kind of pulls you to a leadership position with the organization. And so If you look into the organization, you really like it, you want to get some experience and add something to your CV, you can apply to be on one of the committees for APSA. So they accept undergrads, grad students, med students. You can see kind of this little checkbox here. You can check what year you are in training, and they accept all types of people to be on the leadership committee. So you can help put together these application interactive sessions. You can help organize an annual meeting. There's just a bunch of different ways you can serve as mentors, put together research, virtual summer research programs for people underserved in science and medicine. It's just a great opportunity to kind of get your feet wet if you're interested in physician scientist things. Okay, so now I'm going to talk about MD PhD programs, because I think I didn't learn about them until I was late in my undergraduate training. So I don't know who already knows about them and the structure of them, but this is just kind of an overview of kind of what MD PhD programs are. So this is the way my program was organized. So you have your first 2 years of med school, and then you have 4 years of graduate school. So it's kind of sandwiched right in the middle of med school here. So the PhD part, and then you have your last years of med school. So here, for anyone who doesn't know, I'm going to break down kind of what happens in each of the years too really quickly. So your first years of med school are kind of this classroom-based learning. So there's usually about 200 people per med school class, 200, 250. You all come to these big lecture halls, usually from 8 a.m. till noon. You're just in lectures from that time, and then the rest of the day you get to go study or do small group sessions or etc. So the first 2 years are really just come, sit down in a lecture, learn the material, and then go study. And it's a great time because as a class, you're all taking the same material at the same pace at the same time. So you all kind of take your tests at the same time. So if you're studying immunology, say that's 3 weeks worth of material. So you sit down, you study immunology with everyone, you listen to the lectures, you study it at night, you take quizzes, and you take this big immunology test, and everyone's taking on the same day. And then when you're done, everyone gets to celebrate together. So we used to rent out like spaces and do like karaoke or something fun, like with the whole class. So like 200 people that are really going through something together, and it's just a really fun time. And yeah, it's challenging because you're like going at this really fast pace. It's a little different than undergrad as far as like the volume. But it's just great to have a support system, all these people, and everyone becomes friends. It's like, from my experience, it was such a supportive environment. It's not as like competitive almost as sometimes pre-med can be. So people are sharing notes, sharing information, tips, tricks, all of that. Like everyone's just a big group of friends. So it was a really fun time. And then grad school is when you're working in the lab. There's other types of PhDs you can do. My PhD was in neuroscience, working with kind of cells in a lab and animal models. So I was kind of doing this thing where I was working with cells and dishes of cells and working in like a hood like this. But you can do PhDs in anthropology if you want. You know, there's a lot of different PhDs you can do even in an MD PhD program. And then during your PhD time, you're also kind of writing papers, you're presenting data, you're going to meetings, you're doing all that kind of stuff. And then you finally kind of wrap up with a big dissertation at the end. And then you get to go back to the last 2 years of med school. So your third and fourth year. And so this is when you're finally in the hospital, and now you're kind of actually working with patients. You're wearing your little white coat and have your stethoscope, and you get to go on these rotations. So third year is usually divided with rotating through a bunch of different specialties. So I think most schools across the US, they all have to rotate through kind of the same specialties, and it's neurology, surgery, OBGYN, internal medicine, family medicine, psychiatry. I think that's all of them. So you get to kind of get your feet wet in all these different specialties, really try out what you like and what you don't like, figure out what you want to do. It's a great time. And so that's when I figured out that I wanted to do neurosurgery. So I did neuroscience research. I was always interested in the brain since undergrad, but I was kind of torn between neurology and neurosurgery, and they sound similar, and they are similar. You're studying the same organ, but the day-to-day of both of those is very, very different. And so I really needed to figure out, did I want to be in the OR every day and doing surgery, or did I want to be doing neurology? So this is a picture actually of our dean of our medical school with some students walking behind him in the hospital. But he was actually a neurologist, and I worked with him a lot. But yeah, this is kind of what third and fourth year looks like. You go see a patient, you write notes, you do an exam, you take it back to an attending, and then you all go see the patient together, and you just learn that way. And so for anyone who doesn't know about the hierarchy of med school, because again, I didn't know this before I started, but there's the med students, and then there's an intern, which is kind of the first year resident, and then there's residents above that, and different residencies have different like programs. So 3, 4, 5, 6, 7, all the way up. The last top resident who's been in the program the longest is the chief resident. So they're kind of running the show now. And then right above them is the attending. So the person who's finished all of their training and is now kind of the top of the pyramid of all these people who are working on the service. So this was the way my program's oriented. I'm right here at the tail end. I graduate and get my MD next week. So right at the end of this kind of 8-year thing that I've been working on. There's different organizations to these programs. So here's another example of a different MD PhD program organization. I'd say the majority are kind of this top track where they have 2 years, 4 years, 2 years. There's a couple in the US that have this. You do 3 years of med school. So you're doing your classroom learning, some actually in the hospital, and then you do your grad school, and then your last year of med school. There's some that have the whole grad school first. You do your whole PhD before you even start the med school stuff. And then there's some, I don't know if there's an actual program set up like this, but I do know a lot of people who have done their MD and then in residency do their PhD or after they decide I actually want to do research instead of medicine. So bunch of different ways to get to doing an MD PhD. And, you know, you kind of, if you're looking at applying, the program websites will have information about, you know, how their program is structured. And then a lot of them are flexible. So mine was this top track, and we had a student who was in my class who ended up kind of doing a track 2, just the way that his lab stuff worked out. So he did his whole 3rd year of medical school before he did his PhD and then went back to the 4th year. So usually they're flexible too, which is nice. Okay, so this graphic, and this is not to scare anyone, but so everyone told me MD PhDs are long, right? And I was looking at this and I was like, oh, it's 8 years. I love school, no problem. It's not really intimidating to me. But when people are talking about being a physician scientist and it's a long training pathway, they're actually talking about this overarching longer pathway. So that slide I just showed with the 8 years, that's all this little portion of this graphic here. So you're an MD PhD student. Then you have to do your residency. And like I said, that's kind of 3, 4, 5, 6, 7 years. And then you're a young faculty trying to get a job somewhere, trying to work at a academic center. And then you move to being a junior faculty. And then finally, at the end of this, you finally get your first kind of funding. So they call it an R01 grant from NIH, but you get funded and now you have a lab that's running. So it's kind of an overarching long pathway. And everyone talks about how long it is. And, you know, they have this arbitrary kind of cutoff here at the end, like you're a real physician scientist once you get this funding. And so I'm here to argue that I think, you know, the whole time, I don't like to look at it as this whole long training pipe that I'm just waiting to get to the end of. I'm just really enjoying my time now. And I have a couple slides that hope like show you that I've had a blast like during my training. So I did med school in Augusta. This is kind of like what our med school building looks like. This is this map of Georgia and just shows you kind of where Augusta is. So that was my first 2 years. And then I went to Athens, Georgia for my PhD, which is a fun college town, kind of more in the center of Georgia. They just won the national championship in football. It's a big football school. So that was a lot of fun getting into the kind of the football culture. Met a lot of great people here. And then I went back to Savannah, which is where I am now for my last 2 years. So my program was very unique in that you have to travel around like this. But it was great. Like I'm living my life. This is your 20s when you're in these kind of training programs usually. So it's an important time in your life and you don't want to feel stagnant or that you're not kind of progressing as a person and well as, you know, in your professional life. And I've had a great time, and moving around has been really instrumental for me to feel like I'm not like stagnant. So just to kind of hit it home about how much like, you know, I've got to have a life too while in this training program. I've been able to play on intramural teams while I've been doing my training. So this was a bunch of grad students and us playing volleyball. This is just random people from Athens that I met. So I got to make kind of science and medicine friends and not science and medicine friends during this time. Got to go to things like trivia. Got to do some outreach programs. This was before a Georgia football game. We got to put up these tents that are called STEM zone tents. So we would just kind of teach kids walking by about like science and medicine before these football games. So that was a ton of fun too, because it was kind of putting like tailgating with outreach. That's another event we had where we're giving like posters. In med school, we had something called Dr. Prom. So we all would just like kind of dress up and like have a prom. So you're just having fun this entire time. You know, I don't feel like now that I'm kind of at the end of the journey, like I don't feel like, oh, thank God that was over. Like I've just been having a great time, and personally and professionally. So these are just 2 examples of some cool professional stuff I've got to do. So I got to give a TEDx talk, which was pretty cool. And I also have started kind of becoming more interested in art, and I made this sculpture. This is actually a brain cerebrovascular system, so it's kind of the blood vessels that are in your brain. But anyway, I made this sculpture and it ended up on the cover of a journal. So just a bunch of cool, stuff, just to say that, I think if you look at these overarching pathways, like 8 years, 20 years to get through all of it, it can be a little intimidating, but you're living your life this whole time. Like, it's not like you're missing out. So next, I am moving on. Now that I'm finishing, I'm about to move to residency, and I'm going to do neurosurgery residency, and I'm moving to Duke in Durham, North Carolina. So this is just at the bottom here. I just kind of have a little key showing you kind of what residency looks like, specifically for neurosurgery, and then specifically at Duke. So PGY1 just stands for postgraduate year 1, postgraduate year 2, 3, 4. So they have a clinical year, clinical year, clinical year, and then the 4th year of the program, it's 7 years long. The 4th year is research year. And then you have clinical, and then a half 50-50 research year kind of at the 7th year. So this was really important to me, someone who loves research, someone who's done an MD, PhD. I want to kind of stay involved in both the research and clinical stuff. So I really wanted a program that had research built in. And then after this time, you get to do a fellowship, do a little bit of more training, because why not? And then you get to be like an attending at the end of all of this. So I have kind of another 7-year training path in front of me now. But it's going to be fun. I mean, this is a picture of Durham. It's going to be a cool new city. I'm just really excited to do this next phase. And I think it'll be the same way, where I'm just living my life and enjoying it. This is just showing you kind of what a breakdown of a residency might look like. Again, you're not being stagnant during this time. This is the first year. You have all these different rotations. You get to experiencing new things all the time. It's not like you're just doing the same thing over and over and over again for 7 years. Again, there's these 2 research years built in. And then if you're not interested in neurosurgery, or eventually, this is just to show you that there's other residencies that do have research built into. So this is an example of a urology residency, another surgical field, but you do your first 2 years and then you do some research time and then you go back and finish. So they have them for internal medicine and neurology, pretty much just to show you that if you are interested in PhD and MD and all, or just you are just doing an MD and then you get out and say, I'd like to do research too, you can do one of these programs where you get time for research. You don't have to be an MD, PhD to do these training paths. So yeah, something to think about. If you're like, I really like doing research, I like my lab work, but I don't know if I want to commit to an MD, PhD. You can still do research if you're an MD only. So yeah, this is just an example. Okay, so that was kind of like the first half of my talk. And I just want to see where we're at time. Okay, good on time. So this is the second half that's kind of more the science-based stuff. And I think we have questions at the end, so I'll just kind of keep churning on here. But yeah, I'll get into some of the research and then we'll get into more specifics and cool parts about neurosurgery. So I just have a graphic here to kind of describe what a stroke is, because that's what my research was on. So a stroke is pretty much just a heart attack of the brain. So it's pretty much just one of these blood vessels gets a clog in it, either from plaque buildup from a disease or a clot that breaks off and kind of plugs up the piping. And so what happens is we can't get blood to that certain area. The cells in that area start to die because they don't get oxygen and all the nutrients they need. And then you get physical symptoms that was, I think some of us are probably familiar with from stroke. So facial drooping, arm and leg weakness. So each of these symptoms is going to be specific to the location of the brain where the stroke is happening. So if you have a stroke in a certain blood vessel, that kind of is called the ACA, that blood vessel controls kind of the areas of the brain, or vascularizes the areas of the brain that control your legs. So if you have a stroke in that area, then you're going to have leg symptoms. So it kind of correlates. And that's why I think neurology and neurosurgery is so cool, because you can look at symptoms and really tie it back to the anatomy and where it's happening and kind of pinpoint things. And I think that's going back to kind of what sparked my interest in it in the first place, from that class that I took all the way back in senior year of undergrad. But yeah, so that's kind of what a stroke is, in case anyone doesn't know. And then these blood vessels in this diagram, these are all kind of specific vessels that all have names. And I'm a vascular nerd. Like, I think I should do the vascular sculpture I made, and I really like brain blood vessels. So I'll show you more kind of blood vessel stuff. And when we talk about vascular neurosurgery, we'll talk a little bit more about them too. But yeah, so this was kind of what my PhD was researching on, is kind of how to help come up with therapeutics for stroke. So why are therapeutics needed? This is just kind of showing you a map of the United States and where stroke incidence is. So the darker color means people there are having strokes more. So there's an area called the stroke belt kind of in the south, and you can see this kind of just really dark purple here where people in the south are really affected by stroke. And there's some interesting data showing that if you're from the south and you move to the northeast, your risk doesn't change. So it's something about culture or diet or lifestyle, or there's a bunch of different factors that can go into this, and they're still researching why. But you can see kind of when I last updated these slides, it was the fifth leading cause of death. There's about 15 million people worldwide who suffer from a stroke each year. And it costs a lot of money, and it just really impacts a lot of people. And so one of the things about stroke is not only is it costly and impactful to take a person from the location they're having a stroke on an EMS or the ambulance, get them to the hospital, stay in the ICU, etc. But then usually there's months to years of rehab for a person relearning how to talk, walk, use their hands, etc. And so then there's this like rehab, and it's just a long, costly process. And that's for the people who do survive. So it's a pretty devastating disease. So in my PhD, I worked with an animal model of stroke, and we actually used a pig model. So if anyone's familiar with undergrad research or any research, you probably have seen that people use rats or mice a lot in research. And I just kind of have a head-to-head here about human brains versus rat brains and rodent brains. So humans have a gyrocephalic brain. So pretty much all that means is that it's got these folds. So if this is this human brain, looking at it from the side, this like where the eyes are in the front, this is the brainstem, cerebellum down here. It's got all these folds in it. And so that's kind of a evolution that happened to put all that surface area and all of these neurons somewhere. It kind of just went down in these folds and sulci, and each of these little folds and gyri. The gyri are kind of like the parts coming up, and the sulci are the areas that dive down. But each of these have specific names and specific functions. But if you look at rat brain, this is a rodent brain over here. It's not to scale, it's a lot smaller than this, but this is kind of the front. This is called the olfactory bulb. So this is where all the smelling neurons are for rodents, because smelling is really important to them. So it's a huge, big structure in rodents. Our olfactory sensors are like not even shown. They're under this little, like a little teeny nerve down there. So pretty different already in the organization and size of some structures. But then you can also tell that the brain is smooth, right? It doesn't have the folds like the human brain. And so that has implications for kind of the way the brains are organized, how connections are made. And if you're studying rodents and you're trying to test a therapeutic, it might not be recapitulating what could happen in humans, because they're pretty different. Rodents have only 10% or less than 10% white matter. So you might have heard that there's white and gray matter in the brain. The white matter is the parts that are the connections between kind of the cell bodies and transmitting it to where it needs to go. So that has implications. So if you have a stroke, if the areas that are connecting a higher brain area to what controls your feet is affected, the actual connection part, that's the white matter. And it's important to look at the actual structure when you're looking at a stroke, and rodents just don't have the same kind of structure there. And then lastly, the rodent brain is 650 times smaller than a human brain. So there's all sorts of implications if you're trying to make a therapeutic and get it to a certain location, and the therapeutic needs diffusion to get to where it needs to go. If you're looking at something that's the volume is that drastically different, it's going to take different amount of times and doses, and it's hard to just kind of do a one-to-one correlation. So pretty much pigs, this is an example of a pig brain here, and you can hopefully see that it kind of fits really nicely in between a human brain and a rodent brain. It's kind of fills this gap. It's gyrocephalic, so it's got all the folds like a human brain. It's got more than 60% white matter like a human brain. And even though it's smaller than a human brain, it's a lot closer in size to the human brain than the rodent brain. And then lastly, at the bottom here, these are histological sections of a brain. So pretty much if we took a brain, sliced it this way, and are looking at it, this is a human one. So these are kind of what are called ventricles, these fluid-filled spaces in the middle of the brain that have cerebral spinal fluid, and there's usually spinal fluid all the way, in each of these little sulci, or the areas that dive down. And then you can kind of see the difference in colors where it's a lot darker on the outside, the darker purple, that's the gray matter, and then you have the white matter kind of in the middle here. So you can see pig brain, kind of similar, right? Like it looks pretty similar. You got these white matter tracks like the human brain. And then we look at the rodent brain. And these aren't to scale, so the rodent brain is like way smaller than this, but the same kind of like cross section, but you can already see it looks completely different. It's smooth, doesn't have the same kind of architecture, the coloring, all of that. So we worked with pigs because we thought if it works in pigs, there's a lot higher likelihood the therapeutic we're testing that it would work in humans. So this is just an example showing you kind of some imaging we could do on the pig brains after they had a stroke. So this is the pig brain looking at an MRI. So if you slice the brain this way and you're looking up from the feet, so almost like slice the brain, flip it up, and they're looking at it like that. So this is the pig. This is where the eyes would be at the front here. This is the cerebellum in the back, and then the spinal cord would come out this way. These are the ventricles where the fluid filled is, all the white. Yeah, and so pretty much just showing you here that we could look at a brain of a pig with MRI before a stroke. It's nice and symmetrical and looks healthy, and then after a stroke here. So this area here is the area that's had a stroke, and it's kind of fuzzy and hazy and not symmetrical, and you can see it's kind of off this midline now. It's kind of swelling on that one side, and this is a month after stroke. You can still see the same brain still has damage a month later that we can detect and measure. And so pretty much this is just showing you that with this model and this research that I was doing during my PhD, we could measure through neuroimaging like MRI and look at all these different kind of measurements to see kind of how the stroke was affecting the pig brain. Okay, so I also wanted to test a therapeutic in my PhD, and the therapeutic is called neural stem cell derived extracellular vesicles, or NSCEVs. It's a mouthful. Pretty much all that an NSCEV is a little packet that gets secreted from cells. It naturally just gets kind of like blebbed off by the cell. And it contains mRNA, protein, microRNAs. It's got some proteins kind of in the surface that stick out here. Yeah, so cells naturally secrete this. And then we were taking them from neural stem cells. So these cells that are kind of regenerative, renew, healthy, taking their packets and then putting them on animals that had had a stroke to see if, people have looked at putting the whole neural stem cell and had good outcomes, but there's some limitations of neurals putting a whole stem cell in. So we were like, let's just take the packet and see what happens. And so that's what we were researching in my PhD. And if anyone has heard of exosomes, microvesicles, or apoptotic bodies, I don't know anyone's research background, but extracellular vesicles is just kind of an umbrella term for all of these little vesicles that get secreted from cells. So pretty much recipient cells can take them up, and then it's almost a way for these two cells to communicate and send a little message with all these proteins and things in it to the cell that's going to take it up. Okay, so with these pigs, we also measured their recovery. So in stroke, like we talked about, so one of the symptoms usually is trouble walking or trouble with gait. So these pigs that have had a stroke, they have similar problems as humans. They have trouble walking afterwards. So this is me and an animal. This is a track that measures the actual mat itself. We walk it down. It can measure the animal's velocity, cadence, stride length, paw pressure, etc. So this is just me and an animal, and you can see it walking down the track here, and it's kind of got a limp. He's not walking as quickly. Yeah, you can just see that he's not feeling great. And that was 1 week after stroke. And then this video is going to be an animal that has got those little packets, the little vesicles, and has been treated. Same time point after stroke. Yeah, so hopefully you can see from the video that there's a pretty big difference between the animal that didn't get treated and the animal that did. And they're just walking is looking better. And so all of that's kind of measured out here and all this data on the side showing you that, yeah, there is a statistical difference in how they're walking with the animal with treatment is getting back to normal faster. Here, it's just, I really just like these videos, so put these in here. But if anyone's done cell culture or research or thought about it, so I worked with the animal models, but then I also worked with cells in a dish. And so this is just an example of, kind of what cells in a dish look like, this little teeny dish. Sometimes you have these little dishes that have multiple wells. So this is an example of if we have a microscope on one of those little wells, and these are cells in the dish. So all of the darker blue is kind of like the fluid around it. But pretty much this is a video showing you what neurons, that's the cells that are in here. The cell body is kind of the round part, and then all these areas that kind of like stick out and reach out to each other are called neurites or extensions. And you can kind of see over time, the cells naturally are kind of clumping up and reaching out to each other and making these extensions. So now if I put those little vesicles on the neurons, we can look at the difference between this view. So it starts out pretty similarly. We see everything's bunching up, sending extensions, but pretty much what you'll see at the end of the video is that these cells make more extensions by the end of the video. So if you look left to right, you can tell that the area of this well of cells is covered more by these kind of lighter blue areas reaching out to each other than this one. So pretty much all that means is that if we put the vesicles on our cells, it encourages them to kind of reach out and connect with each other. And you can see how maybe in a stroke that's helpful. If you have brain and neurons that have been damaged, if you can encourage those neurons to patch back in and connect back, maybe you'll get some function back quicker than you naturally would. And then here is just another video of a different type of cell called mesochinal stem cells. We're zoomed out more here, so the cells look smaller. They're actually bigger, but we're just kind of zoomed out more. And all of these little things that look like fish swimming around, those are actually cells kind of swirling around in this plate. And then here's a video of ones that have been treated with the little packets. And pretty much my PhD was looking at the cell work I did was looking at the difference in the sizes, the shape, the motility, things like that with these cells after treatment. So speaking about different shapes, this is another type of cell called microglia. So this is the immune cell of the brain that gets activated. If something goes wrong in the brain, they kind of get activated. They try to figure out what's wrong, and if it's like a invading bacterial organism, they'll try to gobble it up. Or if it's any junk in the brain, like something that gets a cell that dies, they can like help recruit cells to kind of take care of that. So here's just showing you, we put this kind of thing on the cells that activate them. So these are cells that haven't been activated. These are cells that kind of have been activated, and then these are ones that have been activated and given our little packet. So pretty much what I want you to just take home here is that there's different shapes to the cells, and they change with treatment with this little vesicle. And the color, so the amount of redness or orangey tones is how activated they are. So if they, you know, these are kind of the baseline, there's this guy who's a little activated, but the rest are kind of like more blue. If you put the factor on that really angers the microglia, they get more orangey and red. And then if you put that factor on plus the vesicles, it kind of calms everything back down. So this is the idea that our treatment in the brain, if it's really angry and inflamed, we can put something on that calms it back down, and maybe it gives the brain time to kind of repair without this kind of danger signal. Okay, and so I can not only look at cells in a dish that are kind of floating around, but then I can also look at cells that are in tissue. So if we take a brain after a stroke and slice it, almost like, you know, you're slicing a loaf of bread, you have these multiple slices and you can look at different slices. So this is one slice of a brain. The gray part is the glue, that kind of the glue cells, glia, if you've heard of those, that kind of hold the brain together. There's probably microglia back here. And then the dark black is neurons in this section. So it's been stained with something that makes the neurons pop out. So what I did was I wrote a code that identifies these neurons, kind of outlines them, and then gives us a bunch of information about the shape. So these kind of parameters about their actual shape and orientation. And so I don't know if anyone's done undergraduate research yet, but a lot of times if you are doing this kind of work where you're looking at cells in a brain, usually it's the job of the undergrad to sit there and manually count all the cells. So this code lets the images just come in and it'll automatically count it without somebody having to sit there and manually count. So if you are an undergrad who does manually counting, I'd be happy to give you the codes. You don't have to do that anymore. And if you know a lab or anyone who does that, I'm happy to give it so you don't have to do that. But pretty much this is just a whole big brain. This was actually multiple little images that I took of a brain section. This is kind of the midline. This is the ventricle, that fluid-filled space. This is the top of the folds. Took all these little images, ran them through my code, and then stitched them back together to make this big kind of slice image. And here you can kind of see, hopefully you can see there's kind of like a gray matter. Do you see that kind of difference in color? So now this is the red is showing you where the microglia are in this whole big slice. This is an animal that's had a stroke. This is the area of the stroke. It's like a scar over here, this white. There's a lot of microglia in that scar. It's really active, angry. This is an animal that's had a stroke, but also been treated with our treatment. And you can see there's like pockets of cells that are still surviving in this scar area. So we could look at and run this code on all the animals. So each of these pictures are made up of 300 images. So you imagine if you had to look at an image like this and count every cell in 300 images, and that's just one of these pictures, and then you had to do it for every single picture, and that's only one type of cell. So then you're looking at all the cells. It can take a long time to count all this. So the code just kind of does it automatically. And if we zoom in on a little area here by the stroke, See, this is what each of the little images were. Can you imagine manually counting all the little black specks? I stitched them together. And then we can zoom in on actual cells and see what this cell looks like. This is kind of closer to the stroke. This is moving away from the stroke. The cells are kind of changing shape here. You can see as you move away. And so that's kind of a known property of microglia. They have these big swollen roundness when they're activated. They pull in their little retractions and all the little spiky things that stick out. And so you can see kind of this one that's near the stroke is kind of rounder, not as spiky. The one further away is like that, less activated, so it's less round. And then this is how I kind of represented my data, where I'd have like kind of a histogram of what normal looked like from all this counting. This is the area occupied. So pretty much if you took all this and said, okay, what's the area of the black specks and what's the area of not black specks, and then made a ratio. This is kind of what the histogram is showing you. But pretty much the important point is looking, okay, the area changes when an animal's had a stroke. Green is a normal animal with no stroke. Red is an animal that's had a stroke. So the cells change, they get activated, they take up more area. And then animals with our treatment, it's kind of shifted to the left here, kind of closer to normal, but they are still shifting, to the right. And then if we get right in by the lesion, by the stroke, this is compactness. So how round and circular and kind of retracted in the cells are. So this is a normal animal. It's got this nice little normal distribution here. If you've had a stroke, the cells get more compacted. So they, like I said, they pull in all those retractions. Instead of looking like this, they kind of become this, but they still swell up. So they kind of just become this bigger ball. So that's showing a stroke. Animals here are shifting pretty much to more compact. There are animals treated with the vesicles are not having that same shift. So again, this is an idea showing in our actual brain tissue of our pigs that have had a stroke that the EVs are stopping, our treatment here is stopping the activation of these immune cells in the brain, which could help for the recovery. And then this is just a bunch of ways that I represented all that data for some of the studies we did. So this is just one kind of way I put the data for publications. So, right on time. So now we get to talk about neurosurgery. So if you tuned out that whole time during the research talk, I'm sorry about that, but now we'll talk about some really cool stuff. And if you're not interested by this, then I don't know how you can't be, because neurosurgery is awesome. So I'd be remiss if I didn't include one slide about Harvey Cushing in a neurosurgery talk. So he's kind of the founder of neurosurgery. And the real reason I put this in here is to show you how recent the field of neurosurgery is. So if you look at these dates here, when he's kind of developing things and starting his work, these are all pretty recent. I mean, the field of neurosurgery is only about 100 years old, which in the grand scheme of things compared to other fields in medicine is pretty new. And these are all, people that have gone on to have careers that worked with him and under him. But yeah, I really just wanted to include this to show you that neurosurgery is kind of really fresh in the grand scheme of things. And there were societies that were doing neurosurgery a long time ago, thousands of years ago, because they found skulls that have these called trephination holes, or little holes in them that they would drill. We don't know what the actual problem was. Maybe someone got hit in the head, they had a lot of pressure in their head, and they learned if you drill this little hole, it lets out the pressure and makes the person better. So we still kind of do that exact process today, but we just have more advanced tools. They were using something like hand cranks or a chisel. And so imagine thousands of years ago, no anesthesia, nothing sterilized, you're using like crude tools to chisel in. And these people were surviving after those surgeries because we could tell on those skulls that there has been regrowth of bone after the surgery. So it means that the person was alive for a long time, that the bone was able to regrow. So anyway, people have been doing neurosurgery for a long time, but it hasn't become a field until recently. And then neurosurgery is kind of divided into these little subspecialties. So I'm going to talk about a couple of these and just give you like a little example of what a spine neurosurgeon does, what a peripheral neurosurgeon, what a vascular neurosurgeon does. So I am interested in vascular, so that's probably the type of neurosurgery I'm going to do, and I'm going to talk about that type of neurosurgery first. But first I'm going to do a general overview, and then we'll talk about some of these different little subsections just to give you a flavor of what each of them are like. Okay. So if no one's ever been in an OR before, which I had not until I was in my third year of medical school, so you're fine, it's not too late to start, but this is what a neurosurgery OR looks like. So if you're familiar with them from TV, it's actually a little different than on TV. Neurosurgery ones are traditionally kind of bigger than other ORs because you need a lot of different equipment in here. So this is the operating table. Anesthesia, this is the anesthesia cart. They're usually up at the head here. You've got your, surgical lights that you can move around and pull and get to the angle you want to see what you're trying to see. This big arm is an imaging platform that you can kind of wheel up over the patient's head as you're doing surgery and take an image, and it kind of spins around. So you can get a 3D picture of the patient's head as you're doing surgery. There's screens that show you, this is a 3D reconstruction of a blood vessel in the brain. So they're looking at, this blood vessel, and it looks like there's an aneurysm here at the little tip, but probably during the surgery, they can pull this up and look at the aneurysm they're trying to treat as they're doing it. What's not in this picture is there's microscopes usually in neurosurgery, which I'll show you a picture of that next. But yeah, I just wanted to give you a look at what an OR looks like. And there's a lot of different tools they use. So usually there's anesthesia, the surgeon standing here, residents are standing up here doing, medical students are up here. There's a scrub tech, someone who is standing there and handing the tools to the surgeon. So, in every surgical show when they're like scalpel, that's the person handing them the scalpel, which is funny because a scalpel is like 5% of the surgery, or I mean like 0.5. So like you literally cut, and then some neurosurgeries can last multiple hours. So I've been on 24-hour surgeries or just multiple hours. I think the average is probably like 4 hours or 3 hours, but it's like 1 second. And then the rest of the surgery is not doing scalpel stuff. So it's just funny. Who else is in the room? Oh, and then there's an OR nurse. So a nurse who kind of runs the room. And if there's something in the room, you need a certain tool, a certain type of anesthetic, a certain device, they can run out and go get it and bring it back to the room. So it's a pretty bustling area during a surgery, and there's a lot of people involved that kind of make it work. So this is an example of two tweets that Maryland's neurosurgery program sent out. And this is just to show you kind of what it looks like when there's a patient in the room and all these tools are kind of like around and the surgery is going. So there's a patient actually under all this blue tarp. All the blue tarp is stuff that's sterile that you're not supposed to touch unless you're scrubbed in. So you scrub in, you do the thing that's on TV where you like scrub your hands for a while. You put on a surgical gown and these gloves, you have a mask, you have a hair net, and then you can't touch anything that's not sterile. So the blue is safe to touch. This thing reaching over here is a microscope, so that's got a cover on it that keeps it sterile, so you can touch the microscope with the plastic part. And that's why a lot of surgeons will stand like this or stand like this with their hands, because they don't want to accidentally touch anything that's not sterile. But yeah, so this is a microscope in surgery, and this resident right here is looking through the microscope, and then on the screen we can see what the resident's seeing. So if you're standing back and you're a student and you're not actually like the one looking through the microscope, you can still kind of see what's going on these big screens around the OR. But there's IV bags and all these monitors at the feet, and there's an OR nurse to the left here, and just a lot of going on. The anesthesia is kind of on the other side of this drape. And the scrub tech's probably over here. There's a bunch of tools laying on a Mayo stand above the patient. So it's kind of a busy area. And then here's another picture of a resident actually doing surgery through a microscope using the tools. This is neuroimaging in the background. So we can look through a microscope, but then there's also this really cool kind of, it's not augmented reality, but it's kind of like a VR almost space that maps to the patient's imaging. So pretty much you get a little probe or a wand that you can touch the patient's head. So say we think, or we know there's a tumor kind of towards the front of the brain, but we want to go in at the area of the brain that's going to damage the least amount of healthy tissue. So you can just touch this probe there and move this probe around, and it'll show you on the imaging. So this red is probably a tumor, and it'll show you on the imaging where you are on the brain and probably the shortest area to getting in and taking out that tumor. So pretty cool use of technology here. And I included this picture on the right here because I'm actually in this surgery. So that's not me, but I am standing right behind the person taking the picture. So this was at University of Maryland's neurosurgery team. And I got to rotate with them for in September last year for a month. So that's me and the rest of all of the neurosurgery team there. It's a 7-year program, so there's 1 through 7. There's usually 2 residents a year, 1, 2, 3 residents. There's only a couple programs that have 4 residents a year, but this program has 2. But they're a great group of people. It was an awesome time. If you're interested in neurosurgery or getting involved in the surgical, especially you want to shadow in an OR or anything like that, I'm sure you can reach out to the program there. It's just in Baltimore, so not far away. So in med school, your last year, as you're trying to figure out where you want to go and what you want to do, you get to rotate across the country at different programs if you want. So I picked Maryland and got to go there, and it was a blast. So Yeah. Okay. So now we're talking about vascular neurosurgery, which is the one I like. I just want to see my time here. Okay, great. So we talked about stroke. It was what was related to my research. So there's new techniques you can do now called endovascular techniques, which is you don't even have to open up the brain and go in to do a procedure. You can actually usually just use a artery either in the wrist or in the groin, go in through that artery, send a little kind of catheter and tube up and do different interventions without having to go in through the skull. So if anyone's heard of a heart cath, this is the same thing just for the brain. And they have stents that you can put in the brain, same as with hearts, etc. So this is just an example. There's a clot. So this is what's causing a stroke in this person. This is like a 3D rendering, but they sent this catheter up through the vessels. They weaved up from the groin, probably all the way through all the vessels to the brain. And then they use this little retrieval system to grab the clot, and they're just going to pull it right out. And so this is just a picture showing you what a clot looks like that has been pulled out of someone's brain. So someone who is actively having a stroke, the white area is showing you where there's cell damage and there's an active stroke here, and they just went in and pulled this out. So this is really cool. This is newer technology that they just started using a couple of years ago and have had great outcomes with. So the way it's tied into my research is really cool. That the idea that I could physically go in, someone who's having active stroke, go in there and pull it out and save them from this stroke. But then also use some of my research skills to then give them another treatment that helps with recovery, because they have had some cell damage, but they have way better outcomes here with getting this clot out than some other types of getting the clot out. There's other medicines you can take that kind of break down the clot, but sometimes you can do things in combination. So this is just, it's called an angiogram, but pretty much they injected dye up through the blood vessels, and pretty much you're seeing blood vessels in the brain right here. And this is a blood vessel that kind of dives out and vascularizes this side of the brain. And so you can see there's dye, and then the dye kind of stops here. It's like clogged and blocked, and then there's a little bit of dye here, but it doesn't look like the way it's supposed to. So there's a clot here. They went in, pulled the clot out, and now when you inject the dye, look at all of these branches that you couldn't see before because the piping was blocked. So pretty cool, and that's the type of neurosurgery I want to do. Again, that's just showing you what a stroke is again. Yeah, I don't know if these videos, okay, cool. So this is just a little video showing you of, you know, I'll block his, mute him, but just a little case showing you of them going in and like pulling a clot out of a person. So we're going to skip around here. That's kind of what an older clot retrieval device looks like. They have newer ones now. I'll skip around some more. That's what the newer ones look like. So kind of just this little basket that you go up there and like pull it out. So this is a person who was 6-year-old male, had a stroke. He's pointing to the area where the person's having a stroke. And this imaging, so pretty much the blue is where there's not, the brain's not getting blood. So they know he's having a stroke. And this is what an angiogram looks like in real time. So we'll play that for a second. So you see the dye filling all these blood vessels, filling. And pretty much if you're a vascular neurosurgeon and you work with endovascular neurosurgery, you know what blood vessels are supposed to be where, you know what this is supposed to look like. And this is just showing that it wasn't what it was supposed to look like because something was blocked. So they go in, they put the device in, they put it on the clot, and this is it, a live video of it happening over here. So this is the blood vessel that's blocked over here. They're putting it in, they're grabbing the clot, and they're about to just pull it out. And they're going out like through the carotid now. Yep, and that's the clot they pulled out. And so now in this video, you can see this area of the blood vessel that wasn't getting flow is now getting flow. So that's what they're showing you once they pulled this clot out. So pretty cool. And then I have another vascular video to show you. This is just a diagram. I don't know if this is helpful or overwhelming, but pretty much to just show you the red is blood vessels. The yellow here is the optic chiasm. So it's nerves that run from your eyeballs, they cross over, and then they go back to other areas of the brain, and kind of that's how your sight works and gets kind of put into the thalamus and then your optical cortex and centers. And so pretty much anyway, it's a nerve that conducts your sight. And so all of this area is very, very small and packed with a bunch of important stuff. So all these blood vessels, some of these are the carotids coming up on each side, kind of meet, and then they branch in different ways. And then there's the optic nerves are in here. There's other nerves in here. This is another nerve that controls the way your eyes move. So it's all kind of jam-packed in this little area. So if you have an aneurysm, which is what's diagrammed here, an aneurysm is just kind of a part of the blood vessel that started to kind of balloon and puff up. Because there's either a structural problem with the vessel and it's kind of letting a pocket form, or there was trauma or damage done that has caused this sort of genetic defect. But anyway, you can see that if an aneurysm is kind of in this really intricate area where everything's got to sit all nicely together and not push on each other, it's showing this aneurysm pushing on this nerve. And if you push on the nerve, you can start to damage it. And now this person probably was having problems. They have an aneurysm like this, moving their eyeball around. And that's probably where they said, hey, I'm having trouble with my eyeball. Like, can you take a look at some of my brain imaging and see what's wrong with it? And so here is another type of vascular surgery that neurosurgeons do. So this is a neurosurgeon who's actually at Maryland, who's an MD, Ph.D., great neurosurgeon. And so this is showing you, instead of going in through vessels in the femoral or radial, now we're actually doing a brain surgery where we cut it open and look at the brain. So if you don't want to look at surgery videos, you can look away at this part. It's not gruesome, I promise. It's you're just zoomed up in an area looking at the brain, so it's not like really bloody or anything. But just wanted to throw that out there. They are doing a 3D reconstruction of an aneurysm. This is an aneurysm in this patient. So pretty much what he's going to go in is another type. So you can go two ways. You can go in through the vessel and put a coil in this, or you can go from the outside and clip it to cut off the blood flow to this aneurysm. And once you cut off the blood flow and kind of clip it, then the aneurysm will kind of shrivel up and die back, and it won't be at risk for rupturing. So if you've ever heard of, you're going to give me an aneurysm, or you're going to make like my blood aneurysm blow, or people say all sorts of kind of colloquial things. And so aneurysms can kind of pop, and that's when you have problems because you're bleeding out into your brain, which is not good. So anyway, what we want to do is stop that before it happens. So here's a surgery. We're working through a teeny tiny little window. So this little thing here is like a little teeny scalpel. So you can kind of get a sense for how small this area is. And right now he's dissecting the arachnoid layer of the brain. So there's different layers of casings that surround the brain, like the dura and the meninges, if you've heard of dura, pia, and arachnoid layer. So he's going through the arachnoid layer, and it's called that because it looks like a spider web almost. So that white kind of part. And then here is one of those nerves we talked about in that other diagram. So a big nerve is this part he's kind of touching on right here that he doesn't want to damage. He doesn't want to push on too much, and he's got to be really careful. And then on the sides here, this is brain tissue, all of this kind of pinkish stuff with all the blood vessels in it. And so he's pretty much just going through one of those natural sulci of the brain. So, you know, you have the gyri and then the folds. He's following a fold down all the way so he doesn't have to damage any of the actual brain tissue. And so that's why I think vascular surgery is so cool, because you just kind of follow the brain's natural corridors and you can get to the area you need to get to without disrupting things too much. So he's still kind of cutting down here with these little micro scissors. And the scale here is so small. So this little device is a little suction. So he's sucking away kind of CSF and things in his way. And then he has these little kind of tongs and little micro scissors. So the suction device is probably smaller than my pinky nail, just to give you a scale here. Okay, now we have a blood vessel finally in view. So this area that the sucker is touching on is the blood vessel. That little balloon part is the aneurysm, and then the vessel runs this way. So he's kind of just still digging out, trying to get a good exposure of this blood vessel and the actual aneurysm itself. So this is a nerve. So now he's exposed it. This is the vessel. This is the aneurysm, and it's showing you on the 3D reconstruction right overlaid. That's the aneurysm he's going to have to kind of clip and fix. This is a nerve. So he is getting a good look around the aneurysm to make sure he can see the back of it, make sure he's not clipping on anything important. There's another little vessel that looks like it's coming, maybe diving posteriorly there. He doesn't want to get in his clip, but he's going to now take his aneurysm clip and just put it right on the neck there. Boom. And that's it. And now the aneurysm is clipped off. It can't get blood into it. this vessel looks great, nice and healthy, nothing's ruptured. The nerves are nice on this side. He's looking to behind it to make sure he didn't clip anything else besides the aneurysm, like another nerve or another little vessel. And yeah, it looks great. So, you know, that's like a slick, beautiful, clean, nice surgery. And that's, I don't know, that's like perfect for me. Really interested in that kind of stuff. So we will go to next slide here, and I'll try to finish up these last couple ones. I know we're like running short on time, but I'll breeze through these last couple ones. So neurosurgeons can do spinal surgery too. So this is an example of a spinal cord. This is the column with all the vertebrae. These are discs in between the vertebrae. And then this is where your spinal cord lies, in this kind of white area. And so you can see this disc is kind of pushing out here and pushing on the spinal cord, which can cause all sorts of problems for, you know, you get sciatic pain if you ever heard someone say sciatica. So the discs can herniate and push on the nerves here. So neurosurgeons can go in and do special surgeries that take out this disc and make sure the nerves are okay and get the pressure off them so they can work better. Here's a disc protruding out. This is the disc, this is the spinal cord. This is the head up here, the top. So we're up in the neck. Spinal cord is just coming right down. You can see how much it's being pushed on, and that is not good. So they took this disc out, and then they put these rods and screws to give it some structural stability. At Duke, they're doing like awake spinal surgeries now. So you might have heard of awake brain surgeries. They're doing awake spinal ones now. And so that's for people who don't want to undergo general anesthesia for certain reasons, if they're not healthy enough to, if they just don't want to do it. And you obviously get numbed up and stuff, but they can do the surgery and put all this instrumentation in, and you just wake up and go home the same day. So pretty crazy. There's also neuro-oncology surgery. So this is when you're going in and getting out tumors. So this is an example of a brain that has a tumor under here. And so the interesting thing, I think before I was in neurosurgery or before I was a student, I thought, oh, tumor just stands out. You look at the brain and be like, oh, there's the tumor. It's easy, let's just pull it out. But the problem is a lot of tumors look like normal brain tissue. And until you've kind of done this for a long time and gotten used to it, it's hard to tell what is normal versus not. And so when you're a surgeon, you're in there and resecting, you don't want to be taking away healthy stuff, but you don't want to be leaving tumor behind. So it's hard to kind of find a nice balance. But as I mentioned, we have all sorts of kind of imaging now that can show us, this is an example of what the tumor looks like in this person's head based on imaging. And then these are tracts, white matter tracts that we don't want to get into. And this is kind of the tumor, the tract. So if you're coming in from this way on the person's head, you want to take out the tumor, but not go too deep that you get into this tract, because this tract helps the person do a lot of important things. So you want to be careful and not take too much of certain areas. You've heard of awake surgeries, maybe. This is not usually the norm, someone like playing a violin while they're doing their awake surgery. But I have been in part of some awake spinal or cranial surgeries too. And so usually if you're doing an awake surgery, it's because the tumor or the lesion or whatever you're trying to get at is in a really important part of the brain. And so you want to take a little bit, make sure the person can still talk, move their hands, has sensation, and then you say, yep, nope, still good. They can talk, they can play the violin, and then you take a little bit more and say, still good. And pretty much you're just like slowly taking and making sure you're not in a region that controls something important. And then there's all sorts of cool tools that we're putting into neurosurgery ORs now, like VR, just really cool new stuff. So the horizon, it looks pretty hopeful and really fun and like cool use of technology. And then this is the last one, pediatric neurosurgery. So just a little note on that. So there's a neurosurgeon looking at a little pediatric baby. You can operate on anyway from, you know, newborn all the way to an 18-year-old. A lot of times they put in these little shunts. So sometimes kids are born with too much fluid in their brain, so too much CSF, and you put in this little kind of shunt that literally just takes off the fluid. It runs under their skin and then dumps the fluid in their abdomen, where it just kind of gets naturally absorbed. So pediatric neurosurgeons do that. Sometimes this is an example on the cover of a journal with like a little brain tumor in the cerebellum of a kid. So they can go in and do little tumor surgeries. They do all sorts of stuff. So yeah, it's a cool field, and you get to work with cute little babies. And this is the cutest one by far. So yeah, I think it's time for Q&A. So I'll stop there. But yeah, happy to answer anything, any questions you guys have.
Hello, Dr. Spellicy. I'm Hamza, and I'm a BSDO student with Seton Hill University and the Lake Erie College of Osteopathic Medicine. So to start off our Q&A, what has been the most challenging aspect of your PhD?
Yeah, so I think going into a PhD, I thought, showing up and doing all the research was going to be the hard part, and like, taking care of my cells all the time or taking care of animals. And honestly, a lot of times it was getting used to things not working. That was kind of the hardest part. So a lot of times, you have experiments that don't work and you put months of work into it, and something doesn't work, or the treatment doesn't work, or your cells die, or a lot of different things can happen. And so that's kind of something that you have to get used to with a PhD in research. You just have to get used to the idea that things aren't going to work. I submitted grants that didn't get funded, which, months of writing and work went into that. So I think just getting comfortable with not being successful 100% of the time. And as soon as you accept that that's kind of the possibility that, it's only going to work 10% of the time, 20% of the time, then you can kind of be happy with that.
All right, thank you for that answer. I understand you went through a very long process in the MD PhD timeline. So where do the USMLEs fit in, given there's like a bit of a gap between your medical school years?
Yeah. So the way my program did it is we do our first 2 years of med school, and then we took our step 1 exam. So that's a big exam for anyone who doesn't know. It's like a big exam you have to take as a med student. I took it right after my second year of med school, and then I did my whole PhD. So we kind of got it out of the way before I did my PhD, because you forget a lot. So you don't want to do a whole PhD, be away from medicine that long, and then have to take a test about medicine after that. So I took my first step 1 right after second year med school, did my PhD, did my third year med school, and then I took my second step exam in third year.
Okay, thank you for that answer as well. For this question, I know you have some experience with TEDx talks. So for the students who also want to host their own TEDx talk someday, what do you recommend students do to prepare for one and for them to find a host university to do one?
Yeah, so I was really lucky that UGA, where I was doing my PhD, is a site that hosts them. So I had done some other scientific communication kind of talks. So I was in something called a 3-minute thesis talk competition, where I got up on stage and had to whittle my whole PhD down into 3 minutes and make it easy and understandable for a non-scientific audience. So I think I just got really used to doing these kind of like outreachy talks, and I got involved with a lot of different science outreach organizations in the area I was in. And then someone nominated me to do the talk. So I think it was just from my experience being out. So if anyone's interested in doing something similar, I think really just kind of put yourself out there, start doing talks, volunteering for things, doing science communication things like this, like just doing a random talk on a Saturday, because I don't know, I'm really excited about what I do, and I want other people to be excited and find out their own careers too. So I think just getting involved and doing talks, and then someone nominated me, so I got to do it, and it was a really crazy, interesting whirlwind process. And actually, the day I did my TEDx talk, I had to take an internal medicine shelf the same day too. So I did my dress rehearsal, ran to a hotel across the street, took this like 4-hour-long exam, and then ran back and did my TEDx talk in front of however many people. So it was like a nice instance of MD PhD kind of like slamming into each other.
Thank you. It sounds like you're amazing at multitasking. What motivates you to continue your medical journey and avoid burnout?
Yeah, no, that's a great question. So I've always kind of had a kind of split attention brain. So I like doing multiple things at once. Like if I'm watching TV, I can't just watch TV. I've got to be on my phone or doing something else at the same time. So I think I kind of was naturally had a little bit of multitasking built in. But I think I find happiness in getting things accomplished. So if I say, okay, tonight I need to write this thing for this paper and do this outreach thing and make this presentation and blah, 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 blah. So I will kind of jump back and forth from different kind of avenues, either clinical or medicine, and check off these tasks. And that makes me feel good to get things checked off. So I think just kind of really making lists for yourself. And if you get that kind of dopamine burst of getting something accomplished, it's the same kind of idea as going to the gym. Like, it's like, it's hard to get off the couch and like go to the gym, but when you do it, you feel great afterwards. So I think that's kind of how I feel with doing all these things, keeping myself busy. I get this little dopamine after doing it. And so avoiding burnout is a great question, because I have 7 years ahead of me after I just did 8. And then I have an actual career. Then I have to like get a job. So yeah, it's going to be hard, but I think really like focusing on all those things outside of science and medicine are important too. So in my talk, I talked about all the, friends I've made and games I've played, like intramural teams and all that, just really having a life, whatever you're interested in outside of science and medicine, just keep up with that. Don't give up your hobbies and passions.
Thank you. That's really helpful advice for everyone. And speaking of dopamine surges, what's the most exciting moment of your research?
Well, we already talked about the TEDx a little. So that was like a crazy excitement kind of culmination on seeing all this work we've done come to fruition. But I guess one of the most exciting parts was when I got my first author paper. So I took this idea, made a project, wrote it up, and it got published. And I was the first author. So that was kind of a big day, and it's kind of a rite of passage for people doing research. And that one really, that paper meant a lot to me because I had taken stuff that I learned from med school and brought that to the lab. So my research colleagues who were just PhDs didn't really know about this measurement that we use in the clinic. And I kind of said, oh, let's do this measurement on our pigs. We did it. It had great findings. And so it felt like I was really adding to the team as a MD PhD. So that was a cool moment.
And more about your research, given your statistically significant results in animal models, when do you think that NSC-EVs could be used to treat human stroke someday?
Yeah, good question. So the lab that I worked in, my boss, the PI, has started a biomedical company. And so he kind of had one as we were doing this research, and the company is filed for patents and is trying to get the NSCV kind of patents as a technology. And then they work with the FDA. And so the clinical trial process is about a 12-year process and a ton of money to get it kind of underway, and it goes through different stages. And so I think it's nearing, the first stage. So it does preclinical stages, which means all the animal stuff. And now it's trying to near the first stage where you do testing on a little bit of people to just make sure it's safe. And then you kind of work up from there. So hopefully soon, it's just going to be a long process before, if these do work and make it all the way to the end, it's going to be about 12 years before anyone could benefit from them.
Thank you. It certainly is a long process to FDA approval. What is the most prevalent theory behind why the Southern Belt is most affected by strokes in the rest of the US?
Yeah, I mean, it's a good question. I've seen different models looking at different factors as far as like things like climate, diet, mobility, access to healthcare. That's a big thing in the South is access to healthcare. So a lot of states in the South are kind of at the bottom as far as how many physicians they have per patient. Some of the infant mortality rates are worse in the South than the rest of the country. So, it could be a combination of all these things and just access to healthcare. So if you're not going to the doctor frequently, you're not getting meds to help with your high blood pressure. You're not getting meds to help with your cholesterol. So that makes your likelihood of having a stroke go up. So a lot of it might come down to just access, which is sad and why we need more physicians.
Thank you. And clinically, what are some recommendations in lifestyle for patients who are more clinically disposed to strokes?
Yeah, I mean, that's a great question too. I think there's been a lot of data that shows that just being active in any sense of what that means to you, just a couple minutes a day can really help. So there was a study showing that if you do 20 minutes of exercise, like 3 times a week or something like that, you have the same neuroprotective benefits as someone who runs marathons. So there's like kind of a diminishing return. As long as you hit that 20 minutes 3 times a week, you're getting a great level of protection there. So just staying active, and we don't really understand a lot of why, activity or little kind of things like that actually do help prevent some of these cardiovascular events like heart attacks or strokes. But yeah, there's been data showing that little things like that, just doing something, yoga, going for a walk in the morning, running, whatever that means to you. And for like your older relatives who might be more at risk, encouraging them to kind of do something, whether it's get in the pool and just kind of do water aerobics or whatever they can physically do can help. And healthcare, getting the blood pressure under control, cholesterol under control, all that kind of stuff is important.
Okay, thank you very much for those questions. Our next few questions will be a bit more related to the procedure side of things. So the first one is, sometimes migraines are referred to as vascular headaches. So how much are these symptoms of migraines related to blood vessels?
Yeah, it's a good question. So I'm not as up to date on the research in this space, but I've heard that as well. And so they've also shown that, women are more prone to migraines. Sometimes it's tied to people's menstrual cycle. So at certain times in the cycle, they're more likely to get migraines. So estrogen kind of has an effect too on blood flow, the how much a blood vessel will clamp down or expand. And so it's kind of a bunch of these like interrelated things that are affecting blood flow and hormonal changes that could be affecting and causing the migraine. And so, yeah, I don't know exactly the answer to that, but I have heard that they could be vascular causes. So this is one thing I'm passionate about too, is a lot of conditions that only affect women or certain populations tend to not have as much research into them or been investigated. And so neurosurgery specifically is pretty historically has been lopsided. It's kind of a male dominated field. And so we just need people going into these fields that are interested in studying diseases that affect, populations that they might be interested in helping. So yeah, it's a good question. And I guess I don't know the answer, but hopefully someday we will.
Thank you very much for answering anyways. How do you think you'll be applying what you've learned through your research to clinical application as a future neurosurgeon?
Yeah, so the cool thing for me that worked out really well is that my research and stroke and all that really fits with what I'm interested in the neurosurgery side, with the vascular side. So really understanding what happens to the brain after a stroke, understanding the ways that we can help the brain after stroke will be important for me working in the clinic. So, doing those clot thrombectomies, getting that clot out, and then being able to take care of the patient really well after that. So getting them a therapeutic that works, trying to get them on a nice rehab schedule. So learning more, working with my PTs and OTs to really say, oh, I know that getting people up and out of bed early can help with like the rehab part. So I think, all the things I learned in my PhD about what's best for stroke sufferers and the kind of long-term fixing of the stroke will come back and help. And so I think also if I wanted to study tumor too, so that's kind of going a different direction. I've mostly been concerned with vascular, but I know a lot of lab techniques for how you sequence tumors, how you look at kind of the molecular components and breakdown. So the idea is we could take a tumor from a patient, take it, work with collaborators, break that tumor up, look at the little pieces of it, and then try to figure out where that tumor came from, what caused it, what are the best ways to treat it. So you can use the PhD a lot in the work actually, which is pretty cool.
That was really interesting. One more question about technology. You spoke a lot about technologies like 3D imaging and VR. Do you know if any other potential technologies are being developed and how they may impact neurosurgical procedures in the future?
Yeah, so in vascular, the area I'm interested in, specifically endovascular, where they have that little device that goes up and kind of pulls the clot out. There's a lot of new innovation in that space because that's kind of a newer kind of sect of neurosurgery. So making new devices that can go up and grab clots, making devices that can go up and cure an aneurysm from the inside. So it coils around and there's different type of coils, different strengths, different coatings. A lot of people are using 3D printing now too in neurosurgery. So either if it's for modeling how to approach a surgery, you can 3D print someone's aneurysm. So we showed a picture of an aneurysm and had a 3D model of it just on the computer. But you could also 3D print that model, print all the structures that are in the way, and you can plan your approach better before you actually get into the brain and start doing it. People also are 3D printing for patient education reasons. So you can print someone's aneurysm or someone's tumor and then give it to them and say, this is what your aneurysm looks like. This is how we're going to treat it, and just use it, spinal models, same thing, just to teach. What other technology? There's a lot of cool new tools that they're making all the time. So there's sometimes in the OR, there's representatives from companies that make different instruments or different devices, and they'll be in the OR and they're showing you a new tool and say, oh, try this one, try, for spinal surgery, a lot of time we have different instrumentation that goes in the spine. And so they'll say, oh, try this little cage out, try this screw, try this, and you try different things. So all the time there's new inventions. I think the technology part with the VR, I don't exactly know how that's going to be applied. I don't know if it's just for like learning purposes, but there is a lot of cool integration of technology into the OR.
Thank you very much, Dr. Spellicy. And our last question for today is, how do you manage your work-life balance?
Yeah, it's a great question. So I've been really fortunate enough to be able to do a lot outside of science and medicine. I found that planning is really important for me. So if I say I need to get this done by this day or this time on this day, just really giving myself deadlines is how I get to then enjoy time after that. So saying I'm going to get this paper draft done by Thursday at 8 p.m. so then I can watch TV after or play video games or go to the gym or whatever. So I think just setting deadlines for myself really helped. And the other thing that helps is I just really enjoy what I do. I enjoy school. I enjoy the work. I enjoy neurosurgery, and it doesn't feel like work when you actually enjoy it. So that's helpful too. If you like what you do, the work-life balance is already there.
All right. Thank you very much, Dr. Spellicy. Those are all the questions we had for today.
Yeah, thank you very much, Dr. Spellicy. We really appreciate you taking the time to answer all of our questions and taking the time out of your day to mentor us. And I'd love everyone to please give a warm thank you to Dr. Spellicy in the chat box for this incredibly informative session. Thank you.
Thank you, guys. That was great. And if anyone has any specific questions, they can feel free to reach out to me personally. I can put my anyone wants to reach out and DM me on that. I think that's probably an easy way to contact me. But yeah, thanks everyone. Great.
Thank you so much. Yeah. Now we're going to be doing our closing session with our quiz and future session dates. So the link to the quiz is now live. You need a 70% or higher to pass and receive certification. And you'll see that in chat box now. Now on to our next session dates. These dates will also be posted throughout our social media outlets, so be sure to follow us at TeleShadowing. Our next session will be next Saturday, May 14th at 10 a.m. Eastern Standard Time with our physician mentoring us in OBGYN. And thank you so much everyone for attending today's session. It was a great learning session for all of us. We hope to see you in our upcoming sessions, and this concludes this week's shadowing. Thank you.